Hello, everybody. So first of all, I would like to thank the organizers for giving me the opportunity to talk here, for inviting me to this interesting event. I just want to draw your attention that the next event will be organized in Curie Institute, so in my home institution. Okay, so. So we're talking about networks and maps. So in my talk, I will be actually talking about making maps. We do make maps of cancer cells. So I will be talking about Atlas of Cancer Signaling Network, and well, I will introduce the resource and several applications of the resource. So, but I would like to draw your attention first to the basics. So why do we need to formalize the knowledge? Where is a laser? Okay. So first of all, formalizing the knowledge provides possibility to organize, well, the published data, they're distributed in thousands of papers, and create organized resources. Second, it provides a platform for sharing information between different labs. And finally, once the knowledge is formalized, it can be used by analytical tools for analysis of omics data, network modeling, etc., and etc. So, you know that cancer is a very complex disease. Practically every single signaling pathway is involved at some point of development of the disease. So therefore, the systems approach is needed. So several years ago, we collected, published information about processes involved in cancer and created Atlas of Cancer Signaling Networks. Well, lately, this resource has been updated because you know that cancer develops not only in the tumor cells, but also in tumor microenvironment that's surrounding the cancer cells. So, and there are many players that also coordinate the development of cancer cells. So tumor microenvironment is now added into the resource. And importantly, so metabolism is also affected by cancer. So of course, metabolic efforts were taken already in the field. So there is RECON initiative, and now this network is connected to the signaling network, and then crosstalks and cross-regulations between cancer signaling and metabolics are represented. So how actually we build these maps, these knowledge maps? So the information is retrieved manually from the literature by specialists in this field. And then this information is systematically put into the maps. And these maps are navigatable. These maps have particular structure, hierarchical structure, which is important for data integration and interpretation. And finally, now, in the latest release of ECSN, we covered all hallmarks of cancers. So meaning all processes that are lying behind each one of these 10 or 11 hallmarks are there. And everyone can analyze the data in the context not of particular processes, but the hallmarks of cancer, the group of processes responsible for each one of those. So how do we build the maps, the practical side? So we have to be compliant to the standards. So indeed, we use Cell Designer tool and this syntax of systems biology graphical notation. The maps are represented as process description diagrams. So we represent the biochemical reactions with regulators at the most detailed level, including post-translational modifications, etc. So the maps are very complex, and there is a particular structure of the maps. These maps are converted into SMBL representation, and therefore they are not only nice cartoons, but analytical tools. And the structure of the map is important for modeling. So, of course, once you build a big map, an even bigger map as ACSN2 that contains a complex of maps, cancer cell plus all tumor microenvironment, there is immediately a problem of navigation of those maps, like geographic maps. So therefore, we used a Google API and designed Navicell tool for navigation of biological maps, exactly like if you would navigate geographical maps, with all goodies of Google Maps API, Google Maps tools, including markers, pop-up windows, zoom in and out. Everything is familiar to user. Important additional thing that we implemented into Navisol platform is data navigation, data integration and analysis box, where everyone can upload the data in a very simple tab format, and integrate this data into the maps and analyze the data and visualize the data in the context of the map. I will show you an example. So each entity on the map, so here is a top-level view of the structure of the maps. And then while zooming in, you can arrive up to the level of biochemical reaction. So each entity and each reaction on the map is annotated using this format. And then further information about each entity can be studied. And also the literature can be retrieved by clicking on the corresponding links. Okay, so this is a new brand website of ACSN2. As I said, we have cancer cell and tumor microenvironment. Each individual map is accessible. Also, the seamless network of ACSN is accessible. Everything is talking to everything, and we keep this principle that we represent all connections together. Of course, it's an open source, so everything is downloadable in different formats. And as I said, the content of the map can represent signatures of hallmarks of cancer, but also signatures of different players in cancer, like different cells from tumor microenvironment. In addition, the maps themselves, not only gene lists, but also the map themselves can be downloaded, and then the structure of the map can be used for any research. Okay, so this is a multi-scale resource. And this is how the different players are represented graphically, top-level view. This is an example zooming in on one of the maps. So regulated cell death, we collected all possible informations about the way the cell can die. So it's not only apoptosis and necrosis as the textbooks teaching us. There are much more modes, and here they are represented on this map. So the map has a hierarchical structure. It has 3 layers: initiation, signaling, and execution. And then these layers are divided into functional modules. Functional module is a set of pathways, signaling pathways, that guide a particular biological function. Okay, so to sum up the description of this resource. So ACSN covers all hallmarks of cancer. It's manually created from the literature. It's, of course, up to date because we just updated in this year. It's multi-scale and multicellular system. It's built using the standards. It can be then navigatable using Google Maps engine. And it has a hierarchical structure, which is important for visualization of the data. So this is some statistics about the content of the maps. All this is available on the website, so I'm not stopping on this slide. So what is the added value of ACSN? Why ACSN and not others, like KEGG, like Reactome, et cetera, et cetera? So the added value of this resource, it's cancer-specific. So when we compared the literature and the journals from which we retrieved the literature to compose our resource, the body of references is coming from specific journals. So oncological and immunological, those that describe processes implemented in cancer and in tumor microenvironment. Immunological, because the majority of tumor microenvironment is related to immune system. And so it's really cancer-specific, tumor microenvironment-specific, and also, well, the age of the literature, of the references is younger than, for example, in Reactome, because we updated this resource recently. So now let's stop on tumor microenvironment. As we said, there are several players all talking between them and to the tumor cell. Here they are on the maps. We built the maps specific to each one of the players in the tumor microenvironment, shown here. Now, how these cell type specific maps can be used for analysis of data. So we are talking about different players in tumor, but actually when we retrieve a biopsy from a tumor, all cells are there together, right? So it's one biopsy, and we have tumor cells and tumor microenvironment cells. How to separate them? Normally it's impossible and we need to apply particular methods to deconvolute the data. Therefore, we developed a method, it's called independent component analysis, that allows to deconvolute omics data retrieved from bulk tumor samples, and according to the markers, to allocate components to particular cell types within the data. Using this method, we can retrieve components, and then we need to interpret them, because normally components is the only list of genes that deregulated in one sample comparing to another or to normal samples. To retrieve biological information, we perform a gene set enrichment analysis. We developed a method which goes a bit beyond. It's called ROMA. So it calculates activity of the functional modules, taking into account not only the list of genes, set of genes within every functional modules at ACSN, but also mutual influences between these genes. So once these activities of modules are calculated using ROMA method, the scores of activities are obtained and can be visualized on the maps. So actually, what we suggest is actually is a particular pipeline. You retrieve the data, you deconvolute the data, you calculate the scores of activities on the modules of the map, and then you visualize these scores on the maps using Navicell on ACSN website. So this is an example. So we studied status of cancer-associated fibroblasts in breast cancers. So although cancer-associated fibroblasts is one type of cells, they're highly heterogeneous. Within this type of cells, there are many subtypes. How to catch these subtle differences between the subtypes and how functionally to characterize them? So using the convolution method and ROMA analysis, and using cell type specific maps, which represent those processes that are deregulated in cancer, specifically in these cells, we are able to characterize functionally particular subtypes. So here I draw your attention to subtype 1, CAF, and subtype 4. What is interesting is that both of them are contributing to tumor progression. So presence of cancer-associated fibroblasts is very bad prognosis normally. However, these different subtypes actually doing it using different mechanisms. So S1 CAF doing it by releasing cytokines and chemokines that inhibiting immune response around tumor cells, by that allowing the tumor to develop, so it suppresses the immune reaction around tumor. Whereas S4 subtype of cancer-associated fibroblasts are responsible for migration and remodeling of extracellular matrix around tumor cells. So these both subtypes contribute differently, for example, to response to treatment. And by evaluating the status of these two subtypes using the pipeline that I just described, one can predict response to treatment in different patients in breast cancer, with breast cancer. This is one example. Now, as I said before, everything is talking to everything in cancer. And so therefore, we integrated cell type specific maps into a meta map of innate immune response in cancer. Each entity on this map possesses the information about cell specificity. But now we represent all processes together, and then this map demonstrates how the innate immune response regulated in different cancers. So what is the added value of this map? So this is shown on this example where we took single cell, this time it was single cell data of macrophages. And then, so we studied using the same pipeline the differential regulation of functional modules between subtypes in the macrophages. And so we retrieved some particular processes that are normally not described in macrophages, but rather in other cells in tumor microenvironment. We found that these modules are deregulated in these two subtypes, and we could classify these cells into anti-tumor and pro-tumor classes and associate particular processes that deregulated in these two classes and explain why one class is pro-tumor and one is anti-tumor. So both cell type specific and integrated innate immune maps contributing for interpretation and characterization of subclasses of cells in tumor microenvironment. So here I listed several latest papers about ACSN. I would like to draw your attention to this one. It's not about ACSN, it's about disease maps. So you can imagine that we are not alone in the field. There are many initiatives that are trying to map signalings related or perturbed in different diseases, not only cancer, but asthma, cardiovascular diseases, etc., etc. So why don't we join our activities together? And this is how this initiative started. So Disease Maps community is very active. It's having regular meetings, and this next one is going to be in Seville in Spain in 2019. So you can visit the website and check it. So the idea is to build exchangeable modules. Imagine a map kindness pathway. So if this model is already built, why don't we exchange it between different maps related to different diseases? And then particular modifications in different diseases can be added, but the core pathway is already built. This is what we call exchangeable modules. And also, well, there are several problems we try to achieve together within this community. For example, studying comorbidities and anti-comorbidities of different diseases. Using a common repository of all maps from different human diseases. So to conclude, I showed to you the resource called Atlas of Cancer Signal Network that can be navigated by Navicell tool. Navicell tool actually serves many other maps, not only ACSN, and any one of you can upload his own map into Navicell and navigate easily and analyze data. So I showed that it's important to represent cell-specific maps and also integrated maps to study heterogeneity within cancer microenvironment, a tumor microenvironment. And then I extended up to disease maps activity, where we want to study together comorbidities and anti-comorbidities of the diseases using the methods I presented earlier. So I would like to acknowledge our team that helped to create ACSN. So small advertisement of a computational systems biology course. So there is a lot of activities on cancer systems biology in Institute Curie, and therefore we run annual course. So next course is going to be in September. And then there is an open position in our team if you liked what I said. Of course, we have much more activities like modeling and data analysis, tool development. If you are interested, visit our website and apply for the position. And by that, I thank you for attention.
Well, if no one have a question, I have one. So how do you validate the maps?
What do you mean by validate? So
like how do you make sure it's actually biologically relevant?
Well, so the map built by specialists from the field first. Second, we have several curators that go through the maps and look at the content. And formally, we also apply a confidence score that takes into account, well, there is a particular algorithm that applies a confidence score on each reaction on a map. So by that, we can trust, well, the combination of these three things actually make us trusting our maps.
Thank you so much. I'd like to introduce the next speaker, which will be Rodney Rothstein from Columbia University.